Hi, my name is Jason. I'm from the Dione and Goodman Labs at Stanford, and today I want to talk about my dissertation work developing a novel optical mechanosensor that we envision will enable quicker and easier studies of neuromuscular aging. So the global rise in life expectancy that started in the 20th century and continues to this day has undoubtedly been a boon for society. More people than ever are living into their 70s, 80s, and beyond. But as this longevity increases, so too does the prevalence of age-related disability and disease. Dysfunction in skeletal muscle is perhaps the most apparent example of this age-related decline because it very visibly affects everyday functions like walking, gripping objects, lifting objects. But muscular tissues throughout the body experience similar losses in contractility with age. Vascular smooth muscle, for example, gradually loses the ability to regulate vascular tone, which can increase susceptibility to hypothermia and increase the risk of systemic diseases like hypertension. Cardiac muscles also gradually lose their strength, which decreases cardiac output, which can potentially trigger compensatory mechanisms such as hypertrophic remodeling that increase the risk of heart failure in the long term. Now, although aging is the primary non-modifiable risk factor for a whole slew of disabilities and diseases, our understanding of its mechanisms is still very much in its infancy. Short-lived animal models like the nematode C. elegans can help us to better understand what factors contribute to aging and test which drugs might delay their onset. Our labs are particularly interested in the worm's feeding organ, or pharynx, because it exhibits many molecular and functional similarities to the human heart. Both pharyngeal and cardiac muscle cells pump continuously throughout the animal's lifespan with a frequency that can exist at a basal rate in the absence of neural input, but can be stimulated by motor neuron activity. In the heart, these contractions provide the physical driving force to disperse and collect blood, while in the pharynx, they enable the worm to ingest and crush bacterial food. In both organs, the timing and frequency of these pumps can be measured with non-invasive electro recordings of action potentials called electrocardiograms and electropharyngiograms, respectively. Now, sustaining a lifetime of pumping puts extreme pressure on the cell's contractile machinery. This includes the sarcomeric proteins that enable contraction, the ion channels and pumps that shuttle calcium into and out of the cytoplasm, and the metabolic enzymes that continuously regenerate the ATP that powers this machinery. The worm pharynx provides a very simple, genetically tractable model in which to study how these conserved factors, which are associated with aging at the cellular level, translate into contractile decline at the organ level. And because the worm only lives for 2 to 3 weeks, these age-related phenotypes present much faster than they do in mammalian cardiac models. Pharyngeal forces are also theoretically much easier to measure than their cardiac counterparts because hearts can only be accessed surgically. Now, I say theoretically because there's one sizable limitation that prevents us from actually performing these kinds of cardiac aging studies, and that's the size of the pharynx itself. In this cross-section, which I've simplified in the cartoon on the bottom, we can see that the lumen is comprised of one central channel and then 3 apical channels that are connected to the central lumen by these narrow constrictions, which I've labeled with the green arrow. When contracted fully open, as indicated in this diagram up here, the central lumen can draw in material that's less than 3 to 5 micrometers in diameter. This is perfectly sized to ingest bacterial food, but several orders of magnitude too narrow to permit even the smallest pressure-sensitive catheters. When the lumen relaxes closed, water and any material smaller than this around 200 nanometer constriction right here gets shunted through the constrictions into the peripheral channels and then out into the environment. This means that Paula, your molecular-based fret tethers would be efficiently filtered out by the worm, unfortunately. Now, because the worm is such a picky eater, accepting only material within this narrow size range, we needed to develop a micron-sized mechanosensor that could essentially trick the worm into thinking that it was eating bacteria, a plan of attack that I've summarized for you as follows. So with these constraints in mind, we set out to develop a sort of mechanosensitive Trojan horse. But unlike a real Trojan horse, this one isn't actually going to hurt the worm. The sensor, which we've termed a microgauge to reflect both its footprint and its dynamic force range, is built in two parts. The first material component that we synthesize are these force-sensitive upconverting nanoparticles. We then use an emulsion polymerization method to embed them in the second material component, this polystyrene microsphere. And this is a TEM image of those nanoparticles here. You can see in the TEM image on the top left that the dark spots are nanoparticles and the lighter gray contrast is the polystyrene. The SEM image on the top right shows that the sensors are comparably sized to E. coli. And we can also section into individual beads using inside the SEM using a gallium ion beam in order to look at the distribution of nanoparticles throughout the volume. And in one of these cross sections here, the contrast is actually inverted from what it is in the TEM image. So the polystyrene is this dense black matrix, the nanoparticles appear as white flecks, and the outer white coating is just a platinum deposition that's necessary to prevent charging and melting under the ion beam. Now, returning to the first component of the sensor, the upconverting nanoparticles, these are a class of ceramic lanthanide doped nanoparticle, which get their name from the fact that they emit a higher energy light than they absorb. In our case, we send in multiple photons of 980 nanometer light. They get absorbed by ytterbium dopants in this core, indicated in the yellow. And then they transfer this energy to erbium dopants, also in the core, which then emit a single photon of higher energy red or green light. We use upconversion rather than standard fluorescence, which is a down conversion method, because near-infrared light can pass through tissue without inducing autofluorescence background. Using a confocal, a coupled atomic force microscope, or confocal AFM, we showed that the ratio between these two emission peaks, the red and the green, increases linearly with compressive force. This dependence also exhibits no hysteresis across multiple compression relaxation cycles between 0.4 and 15.2 micronewtons, which is important for measuring forces inside of an organ that's continually contracting, and you can't know a priori how many times your sensors have been compressed and up to what force prior to measurement. Now, to actually deliver the sensor to the pharyngeal lumen, we simply mix the microgauges with E. coli and plate the mixture onto an agar pad. This enables us to deliver sensors across hundreds of freely crawling and freely consuming worms in parallel without any direct researcher involvement. We also perform biocompatibility studies, such as this brood assay, in order to confirm that consumption doesn't affect their fecundity, pumping rate, or ability to defecate. And yes, you heard that correctly. I have taken many, many videos of worms pooping. Before they've had a chance to do that, though, we can collect these microgauge-fed worms and prepare them for simultaneous mechano-optical and electrical imaging assays. The worms are first injected into the narrow immobilization channel of this EPG chip, electropharyngiogram, and seated between two electrodes. Using a standard inverted microscope, we can excite the microgauge fluorescence with our 980 nanometer laser, and then collect the upconversion luminescence over time. We can then correlate this with simultaneous electrical measurements from the muscles cell action potentials that are actually inducing these compressions. And using these electropharyngiograms as essentially a timestamp, we can localize event-triggered averages from each pump. So in this left panel here, you're seeing the normalized average from a contiguous 60 seconds of pumping in a single worm. And on the right panel, we're seeing a single representative example from one of these 70 pumps. Following the onset of contraction at the upward depolarization spike, we get an increase in the red to green ratio that indicates an increase in force. And then at the downward repolarization spike, which is the initiation for the relaxation of the pharyngeal muscles, it reaches peak contractile force, and then the red to green ratio begins to decrease afterwards. Now, not only are the force dynamics temporally consistent with action potentials that induce them, but the force magnitudes that we measure are sufficient to lyse their E. coli food source. To make this measurement, we first convert the maximal percent change in red to green ratio into a force change using our confocal AFM calibration, and then apply a Hertz model to estimate equivalent compressive stress based upon nanoindentation studies of single microgauge compressibility. The average force increase that we measure from 180 separate pumps across 7 individual animals is 10.4 micronewtons, which is equivalent to an average Hertzian stress of around 84 megapascals. High-pressure homogenization studies suggest that this stress is sufficient to lyse bacteria, specifically E. coli, at a rate of around 99.9%, so a log 3 inactivation. Importantly, this is also the experimentally observed inactivation rate of young adult worms freely swimming in solution, which researchers can measure by feeding them a fixed concentration of bacteria in their medium, measuring the number of bacteria that enter the pharynx in a given time period, and then collecting the live gut loading and measuring the number of colony-forming units. This, along with the action potential correlation data, are essential sanity checks to ensure that the optical response that we measure is truly the result of intraluminal force. And through this proof of concept, we are enabling the next generation of grad students who are not me to embark upon a muscular aging study with quantitative confidence. But that is the story for the next PhD. For now, I'd like to thank you all for listening and to thank our collaborators and our funders in the Dionin and Goodman Lab and our funders, and I'm happy to take any questions.
Thank you so much. We have time for a few questions.
This is incredibly cool. I'm wondering if you see any spatial distribution of forces within the pharynx.
Yeah, so we don't actually look across different spatial areas. We're looking in a specific compartment in the pharynx called the grinder, because it is grinding up the bacteria. And it's only a compartment that's about 10 microns long, a few microns wide. So we're just concentrating all of our signal as the average in that single compartment.
And if I could come back with a second question. Have you tried feeding the worms different kinds of bacteria that might be, I don't know, softer or harder, and seeing if the pharynx responds to those, or if it's just like a single consistent level of force?
Yeah, so I haven't gotten a chance to look at anything but the standard OP50 E. coli and the wild type worms. But that's a really interesting question. You can begin to ask questions about, is there a myogenic response in the worm pharynx? Would something that distends it produce a bigger pre-stress, as you were, in analogous to cardiac physiology, that would end up producing a higher stroke volume or load on the nanoparticles afterwards? So that's something that we're definitely interested in studying.
Do you know what's the temporal response? Is basically instantaneous for the nanoparticles, or if you push them fast enough, they will lag, or?
Yeah, so the process that we use, up conversion, it has a lifetime of around a sub-microsecond, but not very sub-microsecond. So unlike down conversion processes, which can be like nanoseconds, hundreds of nanoseconds. So we restrict ourselves to a 20 microsecond integration time.
Looking ahead to experiments that the next generation might do, as you mentioned, are there any worm, genetic mutants that are known to accelerate aging or preserve the lifespan of the worms that you would be interested in looking at with your system?
Yeah, so the worm has been used for like decades at this point to study aging mutations. So one of the big ones, one of the canonical ones, is the DAF2 mutant, which has a deficiency in insulin signaling. So it's essentially a nutrient sensing defect that affects various stress sensing pathways. And it causes the worm to live for about double the lifespan. It's interesting because within that doubled lifespan, you essentially have a vastly expanded gerospan, so a period of decrepitude that's extended relative to the wild type. And it would be interesting to look at that DAF2 background and then knock down specific components to see which ones confer that longevity without that extended gerospan.
That's so cool. Thank you so much.
Thank you.